else that's required, but just a non-active brain, is it possible to extract memories from that static structure? Now, that's really hard. So we also have each particular year 4 annual research awards for people who make a significant progress on the way to achieving that overall main goal. So that might be coming up with really good new techniques that enable us to image engrams or other aspects of brain structure relevant to memory decoding. It might be for clever new experiments, it can be for all sorts of different things. And those are generally, just to honor and highlight research into how learning and memory are physically encoded in the brain. And so we're going to have another one of these events and hand out some awards in November at an SFN satellite. All right. So I'm going to give some background, as Oge was asking about, for this particular paper. So this paper is interested in systems consolidation of memory. So when we're talking about memory consolidation, what we mean in a phenomenological behavioral sense is that memories tend to become less specific and more GIST-like over time. So you might start with something like, I bought bananas at Sammy's supermarket on Tuesday, and then in a month from now or 2 months from now, it becomes, I remember shopping at Sammy's supermarket once. You sort of distill the gist of the original specific episodic memory down into just some core general components. And historically, it's been suggested that this happens as the dependence of memory shifts from the hippocampus to the cortex. And there's a bunch of reasons for this, but the key one is that if you get hippocampal lesions, these seem to damage recent memories while sparing remote memories. That's definitely in the human psychology literature. It's also reviewed for some animal work in this great review paper by Tonegawa et al. From 2018. But there are some complications. And the other thing is that you can see that prefrontal cortex neurons are the ones that become more and more sensitive to activation by natural cues during remote memory retrieval. So basically that's saying it weeks, a month or two later, if you expose an animal to the natural cues that might trigger its remote memory, you see activity in the prefrontal cortex in a way that you didn't initially see, and you see silence of hippocampal neurons that might have been initially active during earlier recall at only a day or a week after the initial memory formation, for example. The complications start to come in that some people say there is evidence for ongoing dependence of the hippocampus for particular kinds of memory. So some say for discrete, context-rich memories, so episodic memories that aren't very GIST-like, that are still quite distinct memories, those, even when accessed weeks or months later, may have some ongoing dependence on the hippocampus. So for example, there's this paper here from 2011 that showed that inhibiting the CA1 using optogenetic tools, so the CA1 is one of the subregions of the hippocampus, inhibiting that region during retrieval of remote memories impaired its retrieval. So you guys can see my mouse on the screen, right? Yeah. You can see when the light was on, you see this dramatically reduced freezing in these animals compared to control animals, even when tested at 28 days later, compared to when there was no CA1 inhibition. So that's an example of the hippocampus being involved in remote memory recall during contextual fear conditioning. But then there's also some hypotheses that the GIST-like transformation of memories occurs in the hippocampus and not just in the cortex. And that's motivated, I think, as they say in this paper, more by sort of human neuroimaging studies showing hippocampal involvement during GIST-like memory recall. But to be honest, I haven't fully reviewed the literature on what motivates that initial thinking. I just know that there have been hypotheses that is what's happening. So this paper is going to be looking at what happens to memories over time. What happens to memories in the hippocampus, or specifically engrams in the hippocampus over time. So going from 1 day to a few days to 28 days, and seeing the transformations that take place and the behavioral changes that go along with those physical transformations. And there's some real top cast of like engram researchers here with both Sheena, Josselyn, and Paul Frankland, who've been really pushing this field forwards for a long time now. All right, let's get into it. So, and again, just feel free to put your hand up, ask questions as we're going if you want anything clarified. So they use contextual fear conditioning, as do basically every paper that we've been discussing over the course of these journal clubs. But just as a brief reminder, in case you haven't been before, that's a paradigm where you condition an animal to be scared of a particular tone, like a high note or a low note in a particular context. So in one particular cage, let's say A, that might have distinct smells and sounds. And then later on, you can put the animal back into cage A or a different context, let's say context B, and you can see if the animal freezes in just the sort of context where it was initially given a painful, scary stimuli, or whether it's forgotten that, or whether it's overgeneralizing and it's freezing and being scared in different contexts that weren't the same as where it was initially scared. And you can do this for memories that are recent, so at 1 day, intermediate, at 14 days, or remote, so at 28 days after the initial conditioning to be scared of context A. And what they show here with just some control mice to start with is that you see that for recent memories, so for 1 day, animals freeze a lot more in the scary context, context A versus context B. For intermediate memories, they're still freezing a lot more for A versus B. But 28 days later, they're freezing in A, so they remember, in a sense, that A was scary, but they've started also freezing a lot in context B, where they weren't initially freezing back on day one. And you can see another way of quantifying this is the discrimination index, so their propensity to freeze in A versus B reduces over this 28-day period. So what they do is they use this to define what they call a gist-like memory, which is they're saying that the animals have developed gist-like memory when they have equally enhanced freezing for both context A, when they were initially conditioned, and context B, which was initially this neutral context, as opposed to just A. So they're essentially saying, look, the animals have become less specific about where they freeze. They just remember vaguely that this whole A, B, just like different contexts they're being placed into is scary. So that's when we talk about just like in an animal model throughout the course of these experiments, that's what they're referring to. Okay. So having established that, they wanted to know what is going on in the hippocampus during this consolidation period, where they see these behavioral changes. And the way in which they do that initially is they just do some labeling of the cells that were active during the initial fear conditioning. So this is with the standard sort of FOSS-CRE setups we've seen before, where you can essentially use an injection after the conditioning to label cells that were active during the conditioning period. And then you can see either 1 day or 14 days or 28 days later, which of those cells were active during the initial conditioning. And you can compare those to cells that were immediately active just prior to when you sacrificed the animals and imaged them. So the engram cells here in red, labeled with EYFP, are the cells that were active during the initial fear conditioning. And the cells marked in gray are the cells that were active in exposure to whatever environment the animals were in just prior to being sacrificed. So whether that's A or B or home cage or whatever. And you can see here on the right that the FOS plus neurons are outlined by the yellow dashed lines, and the arrows mark FOS neurons, so neurons that were recently active, that were also engram neurons. So there were also neurons that were active, during the initial fear conditioning. So you can see, for example, some of these neurons that are yellow but not red are neurons that were active just prior to being sacrificed but weren't part of the engram. Some, like this one, were part of the engram but weren't active just before sacrifice. So, they weren't used for recall and if the animals were exposed to A or B. And some are both. Like this one in the hippocampus was active, let's say this was at 28 days later, both 28 days ago and also just now, just before the animal was sacrificed. So what they do is they look at whether this reactivity in the hippocampus lines up with the behavioral sort of signatures of remote memory access and just-like activity. And what they show is that in the dentate gyrus, for example, this is looking at the proportion of cells that were part of the original engram, so labeled by EYFP, that are also active just prior to the animal being sacrificed, as a proportion of all cells that were part of the original engram. So this is all cells that are part of the initial engram, labeled in EYFP. And this is cells that were active just prior to being sacrificed and also part of the original engram. So what you can see here is that animals that were sacrificed after day one, so like what they were conditioned, and then 1 day later, they were put back in context A or context B, and they saw which cells were active. For that circumstance, you see a lot of activity of reactivation if the animals are placed in context A, but little reactivation if the animals are placed in context B. So the dentate gyrus is being pretty specific for A versus B. And then 28 days later, the dentate gyrus has no reactivation of those initial cells in either A or B. So dentate gyrus is only being involved clearly in these recent memories, and it's not being involved in these remote memories. This is in contrast to what they see in the CA3 and the CA1, whereas in the CA3 and CA1, at recent time points, they see selectivity for the correct, the initial scary context, A versus B. But at remote time points, they're seeing equal reactivation for both context A and context B, which again is like showing that these hippocampal regions are becoming non-specific over time in a way that lines up with the behavioral data that they showed previously. So yeah, just to recapitulate that, the dentate gyrus appears to have no role in remote engram reactivation, whereas the CA3 and CA1 activity becomes non-specific and just like over time. So then the question they had is, looking at this, they were curious about, well, what's specifically happening with this CA3 to CA1 activity? What's changing not just at the cellular activation level, but at an electrophysiological level that might be involved with these processes? So to measure that, they switched from analyzing just cell activity as imaged by FOS, and instead they essentially labeled these cells with a genetic calcium indicator. So they're labeling the CA3 neurons, and then the CA3 projects to the CA1. So they're placing their fiber over the CA1 so that they can watch the activity of the CA1 neurons as influenced by the CA3 projections. And you can see here some examples of traces of calcium recordings taken. And you can see that for recent, when imaging the CA1, you see this recent activity. There's a lot more spiking this isn't spiking, a lot more events of fluorescence for context A versus context B. But that over time, you see that there's an equivalent number of events in both context A and context B. So this is essentially an electrophysiological signature that lines up with their previous FOS imaging. And sort of quantifying this in a different way, you can see that there's this increased activity just prior to freezing bouts that can be imaged here in the CA1. Or as another way of putting it, these graphs are a little hard to interpret if you initially look at them. But you can see that for recent memories, basically you see these events, these changes in fluorescence, a lot more in context A than in context B, as shown by it's way off to the left of this diagonal line, which would be equal numbers. But over time, it comes back to being more equally occurring in context A and context B, as also, I guess, you can show here in this particular plot. Now, I'm just going to check in. Everyone is still with me so far. Okay, cool. Seeing some
with nodding. You, but still thoroughly confused.
Yeah, great.
Yeah.
As long as we're confused with the paper instead. Sorry, Ken?
One quick question that I think I know the answer to, but it might be relevant. The usual order is that information comes into dentate gyrus, that goes into CA3, then goes into CA1. Presumably, what they've shown here is that dentate gyrus is not the thing that is kind of involved in this. Do they say
dentate gyrus isn't the only path in? I mean,
okay,
you've got the entorhinal layer 3 and 2 going into CA3 as well.
So CA3 directly.
And so there are other paths, but dentate gyrus, I mean, I guess. Sorry, I'll hold off on further interpretation because I'm just slightly confused. Although I could, no.
There'll be a lot more on dentate gyrus contributions to this activity as well, Ken. So probably better we hold off on that. They're at least showing that they don't see the same GIS-like changes or lack of selectivity changes occurring in the dentate gyrus like they do in the CA3 and CA1 at remote time points. But yeah, there's a lot more to come on the dentate gyrus. And then lastly, so they've shown these changes in selectivity of the CA3 and the CA1 over time, where the CA1 and the CA3 are becoming active for both context A and context B instead of just context A at these remote time points. So they want to check this a little bit to see, well, what happens if they block this CA3 to CA1 activity at these distant time points? Like, does that affect freezing? Can the animal still freeze? Do they require the CA3 to CA1 circuitry for memory recall at these distant time points? And this is kind of interesting in that this is related to those lesioning studies that I talked about briefly in the introduction, but instead of like lesioning out the entire hippocampus or totally lesioning out a like full subregion of the hippocampus, they are just blocking that CA3 to CA1 engram projections. And what they show here is that the blocking this circuitry leads to partial but not complete blocking of the freezing activity. Where what they do is they demonstrate that the memory is still there. So they take the animals, they condition them, 28 days later, they re-expose them to context A and context B. They see that they're freezing equally to both context A and context B. Then they apply the optogenetic sort of silencing of the CA3 to CA1 circuitry, and they remeasure how much the animals freeze for A or B, and they see that they get a partial but not complete reduction. I find this kind of interesting in that I think it gets into what we're going to be discussing later, in that they show some degree of behavioral dependence on the hippocampus for this behavior. But they also show that, you know, it's not like the recall of the memory completely goes away based on this CA3 to CA1 blockade. So presumably there's something else that's subserving that behavioral recall as well. Yes, Ken?
Was this bilateral?
That is a very good question that I should absolutely be able to answer off the top of my head, and that I don't remember. Yeah, that's a good point. I forget if someone wants to check that, but otherwise I'll check that and we can come back to it later. All right. Okay, might be actually a very simple explanation, but moving on. What's happening to this circuitry over time? like where are these changes coming from? What's actually happening at a neurophysiological level? So the way they're going to go about doing this is through the eGrasp system that we have talked about again a few times during these various journal clubs, in particular when we were talking about the Choi et al. 2018 paper. But essentially, this involves injecting or micro-infusing a couple of different tags into the animals such that there's a different tag in the pre-synapse and in the post-synapse, such that when those two come together, you get a particular fluorescence tag that marks where these synapses are occurring. So what they do is they inject the pre-synapse in yellow and the post-synapse markers in red, either into both the dentate gyrus and the CA3, so pre-tag into DG and post-tag into CA3, or they do the same where the pre-tag goes into the CA3 and the post-tag into the CA1, so that they can observe what's changing in these connections between the dentate gyrus and the CA3, and between the CA3 and the CA1 over the course of this 28-day period. So they initially label these regions, then they do their context sphere conditioning, then they induce the tagging immediately after conditioning, and then they wait either a day or 2 weeks or 4 weeks before they do their histology. And I've put this in here because this is their little diagram sort of the circuitry that they are monitoring. And they're going to use this diagram a few times throughout the course of the paper to sort of flag the particular area that they're interested in. Yes, I think that's all I wanted to say here. Okay. So what they're doing with these next figures is they're showing off the structural changes that occur over the course of consolidation. So when they're comparing what's happening at the 28-day mark, let's say, compared to the 1-day mark, for both the dentate gyrus to CA3 connections and the CA3 to CA1 connections. So we're starting with the dentate gyrus to CA3 connections. And what you can see here in cyan is a dendrite of the CA3 neuron. And in purple is marked the sort of large mossy terminals, or the connections from the dentate gyrus onto this dentate gyrus CA3, or the connections from the dentate gyrus onto this CA3 dendrite. So the dendrite in general marked in cyan, specific connections marked in purple. And they sort of check a few different things, comparing the recent time point to the remote time point in this imaging, where they see that initially, they see that when comparing recent to remote time points, they don't see any change in the density of these large mossy terminal connections. That's for, large mossy terminals, so from those excitatory neurons in the dentate gyrus onto the CA3. They're also interested, though, in the connections between interneurons and these excitatory neurons, so between these parvalbumin interneurons and the CA3 neurons. So what they do show changes a bit is the dentate gyrus engram filopodia, so these connections between the dentate gyrus engram cells and the CA3 interneurons seem to change a little bit. And to quantify that, they see the number of filopodia decreasing from the recent time point over to the remote time point. So there's a little bit less drive going on to these sort of inhibitory neurons, these parvalbumin neurons, from the engram cells in the dentate gyrus onto the CA3. So those are the dentate gyrus to CA3 changes that are occurring over the time course. No changes in the density of neurons or the positioning of these dentate gyrus to CA3 synapses, but a bit of a decrease in this interneuron connectivity. That's quite distinct from what they end up seeing in the CA3 to CA1 connections, where again here you can see a CA1 dendrite being imaged in cyan, and the connections from the CA3 to the CA1 being shown in purple. And what they show here is that in contrast to the dentate gyrus to CA1, the dentate gyrus to CA3 data, they see a big increase in the density of engram synapses. So the number of synapses goes up at remote time points. And they also see a change in the positioning of those synapses. So they seem to cluster up over time, where they start out more spread out, and they're becoming clustering at the remote time points, which they can quantify with this sort of nearest neighbors index, where you see we've gone from like 8,000 to 20,000 synapses. And those synapses are also a lot closer to each other. So they've gone, what is the distance? I think this is in micrometers. So they've gone from an average of like 5 micrometers to an average of like 3 micrometers or something in distance. So these are the structural changes that are occurring over the course of this consolidation period. Yes, Ken?
Sorry, I want to double check. So the only synapses that are shown here are the ones that are labeled with egrasp. So by definition, the cell that we're looking at is an engram cell that was labeled at the contextual Führer day, and the CA1. I'm sorry, the CA3 axon that's making these was also labeled.
Yes, that is correct, except for these parvalbumin interneurons are, I believe, just labeled using PV. It's still an engram cell that's projecting onto them, but the interneurons didn't have to have egrasp in them to be labeled. But otherwise, everything else has egrasp labeling, yes.
Okay, so when on this slide, when you're showing that the density of engram synapses went up 28 days later, that is still from the same. So new synapses. The only way that this works is that new synapses from those original CA3 engram cells had to form on this.
Or there are new connections that lead from, say, the recognition of context B to the engram cells in CA3 that were involved with context A.
That's a good point. So it might be that they're okay, but it's still an engram cell.
Sure. Yeah, it's still the engram cell, and these were both involved in the original context, right? In context A. That's how they were labeled, I assume.
Yeah.
Yeah. So this CA3, CA1, all the synapses that we can see now have to have been part of that, and now more of them are becoming involved in remote memory B. So there has to be a way that B is then pointing to the original synapses involved in engram A. If I'm understanding correctly how this labeling happened.
In any case, you answered my question. Thank you.
Yeah, that is also my understanding, as Randall's put it. We can dive into that a little bit more once we go through the data. So to continue on, so they're interested in these changes, and because it's the hippocampus, they wanted to know whether neurogenesis is affecting these things. At least that was my understanding. I didn't get a 100% clear view on why they initially thought neurogenesis would be critical for this, or whether they were just interested, but nonetheless, they investigated its role. I guess because historically neurogenesis is thought to be important for pattern separation, so making different memories distinct, whereas this is kind of looking at making memories more just-like and less distinct. So I'm just curious about what their original motivation was, but It's not clearly outlined in the paper. Nonetheless, so what they go about doing in this next section is they're doing the same sort of labeling as before with the e-grasp of dentate gyrus and CA3, but this time they're also adding in a step where they're adding in a label for newly formed neurons. Where what they're doing is they're labeling the newly formed neurons, and then 2 weeks after they label these new growing neurons in the hippocampus, they do their fear conditioning, and then they wait to do their remote sort of behavioral assessment and then histology. The reason why I'm clear about this being 2 weeks between when they do the labeling of the new neurons and the fear conditioning is because that's not enough time for these neurons to then be functionally integrated into the dentate gyrus when the actual fear conditioning happens. So 2 weeks later at fear conditioning, there's still going to be an engram that's going to form to correspond to that memory of A, but those new neurons won't have been involved in that engram yet. They're only going to get integrated into the hippocampus properly over the next couple of weeks during the consolidation period. And that's kind of key to distinguishing some of the results that will be seen in this paper compared to in other papers that they're talking about. Yes, Randall?
But, okay, so you're saying they're not mature, they're not fully involved in engrams at 2 weeks, but the way that a neuron matures and how its, how its dendrites and axons grow out is influenced by activity. So that could influence what they end up connecting with. I'm not entirely sure that this really separates the two.
That is true. I'm not fully on top of the process by which these newly born dentate gyrus neurons start to become integrated and when they have any degree of integration into circuitry and how that occurs. I'm just flagging it because I distinctly remember, and I think it mentions in this paper as well, that like full-on functional integration happens at like the 3 to 4 week mark, which is different because sometimes they show, for example, that if you, and as they will show in this study, that if you ablate these neurons, you get behavioral changes. And this is to point out that if you ablated all of the new neurons and then you measured something like a day later or 2 days later or 5 days later, before those new neurons had become functionally integrated into the hippocampus, you would expect not necessarily the same changes as if you depleted all those neurons and then were measuring, a few weeks later when those neurons were supposed to have been functionally integrated in. It's really just to point out that the time course matters of like when these neurons are becoming integrated in. All right. So here's kind of a complicated visualization of what is happening at this time point after they've labeled the new neurons, which have been labeled in, again, cyan here. They've labeled the dentate gyrus engram sort of axons in yellow. They've labeled the CA3 engram dendrites in red. And they've labeled the recently active, so during the recall just prior to sacrifice neurons in white. And what they're just showing is a comparison here between, on the bottom, you can see active CA3 engram neurons. So neurons that were part of the initial engram that are active again at this remote recall. And you can see that these ones here, as shown here, have been contacted by this newborn neuron. So the CA3 dendrite and the purple marking the dentate gyrus connections onto it have now been joined by this newborn neurons sort of connection, just immediately proximate to those dentate gyrus, the CA3 connections. Whereas you can see on the top here, this is an inactive neuron. So the CA3 engram, the CA3 cell was initially active during engram formation, but it's not active in this remote time call. And in this image, it's also not being connected to by this newborn neurons axon. Obviously, this is just like one image, so it's anecdotal data. This doesn't quantify anything. It's just a visualization. But if you start to quantify these, what you can do is you can compare the proportion of original engram neurons that are active during the remote memory. So that's what you can see here on the right, which is that at this remote memory time, when looking at context B, you can see about a third of the cells of those original engram neurons are active in context B. So, most cells that were originally active aren't being active for context B, but a substantial proportion, this third, are. But of these cells that are active, you can see that the proportion of CA3 engram neurons with contact from these newborn neurons, so like these kinds of cells here, most of those ones are becoming active during context B. So there's a big discrepancy where like most of these cells aren't active again in context B, but of the ones that are active, most of those have had some inputs from these newborn neurons, which is interesting. It superficially, this makes a little bit of sense to me because I know that these dentate gyrus newborn neurons tend to be a lot more active than sort of the older neurons. But that's just to flag that's the sort of structural change they're seeing here. The question then is as well, like, what happens if you mess with this? If you either turn down or turn up the amount of neurogenesis that's occurring. So they're going to do some interventions and see what happens. The two key interventions that have been used historically and that they use in this particular study is either they ablate the neurons, so they irradiate them where radiation selectively kills off the replicating cells, or they promote neurogenesis. This one's a little bit stranger, but doing exercise, particularly voluntary exercise, is linked to strong growth of new neurons in the hippocampus. So they're able to use that to show a large increase in this neurogenesis process. And they see what doing each of these does to the structural and behavioral results that they've been seeing. So basically, they do these activities 2 weeks before the contextual fear conditioning, and then they do the same sort of imaging. I didn't postulate this out, so we'll just go through it from left to right. Sorry. So again, they're looking at the CA3, the dentate gyrus to CA3 or the CA3 to CA1 circuitry. And this is initially looking at the ablation. So we're looking at neurogenesis ablation. So this is where we've irradiated the hippocampus and we're not going to have these new neurons in the dentate gyrus. What is interesting in this circumstance is they see that fully blocks the GIST-like changes behaviorally. Sorry, that's shown here. The GIST-like changes that happen behaviorally over the course of going from recent to remote. So previously, we saw that for recent memories, there was a lot more freezing in A compared to B. But in remote memories, there was equal freezing for A and B. But in this condition, you see that it's actually remains specific at 1 day, at 2 weeks, and at 4 weeks. So in a sense, it's enhancing the animal's selection of like the appropriate context by removing these newborn hippocampal neurons. And you can see the same thing happening with the reactivation of cells. So the engrams themselves stay more selective in both the dentate gyrus, the CA3, and the CA1 over this time course for remote recall. Interesting as well is that you're still seeing this in the dentate gyrus, whereas previously, by 4 weeks, you weren't really seeing much activity in the dentate gyrus at all, and certainly no selectivity. So they're showing this, the GIST-like changes are prevented at a behavioral level, at this engram reactivation level. They're also showing that you're not getting these changes in palvalbumin interneurons. So the normal decrease in the number of filopodia onto the palvalbumin interneurons doesn't change in the irradiated animals. And also, you don't see a change in the structure, the clustering of the synapses in the CA3 to CA1 connections. So they stay sort of broad instead of clustering up. Essentially, they're showing that irradiating these neurons, getting rid of them, just totally blocks this change from a more specific kind of memory to a more gist-like kind of memory. And then that's in contrast to what they show with neurogenesis promotion, with this sort of running context, where you can see when they are doing the opposite, they're encouraging the animals to run 2 weeks prior to this activity, this imaging. You can see on the bottom here that it accelerates the gist-like changes to these memories in a behavioral sense. So now, even by 2 weeks, they're seeing that the animals aren't selecting context A over context B for freezing, let alone at remote time points at 4 weeks. They're seeing that the non-selectivity of the CA3 and CA1 in those engram reactivations is now occurring at 2 weeks instead of just at 4 weeks in the CA3 and in the CA1. So this is intermediate now instead of remote recall. And they basically see an acceleration of everything else as well. So the changes to the interneurons and the changes to clustering of the synapses as well. And then as their last sort of add-on to their data before we get into the discussion, they repeat the sort of calcium imaging that they did previously, but now with this either neurogenesis ablation setup or this neurogenesis promotion setup, where if you remember from before they did these interventions, they showed that over time, that initially there was more electrophysiological activity for context A versus context B in these connections, but that became unselective over time. And now they want to see if the same thing, like how increasing or decreasing neurogenesis affects that. And it goes pretty much the way that you would expect, where if you, wait, hold on. Am I getting confused here? Let me visualize this. This is event ratio. This is showing irradiation. Yeah, no, 1 is here. So I am getting confused. It does go the way that you expect, where it's just that irradiating the neurons maintains the selectivity of this calcium imaging for A versus B, whereas increasing the neurogenesis removes the selectivity quicker. So it's, again, just the same results sort of as before, but shown in a functional sense instead of just a structural sense as well. So then that's it for data. We've got a few minutes left where we can discuss some of the implications. But as their sort of like final visualization of what they think is happening, what they're showing is that for recent episodic-like engrams, you get these engram cells in the dentate gyrus and the CA3 and the CA1, where you get these projections from dentate gyrus to CA3, which project onto these excitatory neurons and these parvalbumin interneurons, and then from the CA3 onto the CA1 into like broadly touching many dendrites, many areas. And then as you have consolidation occur over this period, you get an addition of newborn neurons which contacts these previous engrams that have been formed and that influence them to a degree. You get these changes in parvalbumin interneuron connectivity, and you get this clustering of the CA3 to CA1 connections, where the dendrites, the synapses tend to cluster together over time. I think this is just me saying what I just said, but written out. All right. So let's get in the last 10 minutes into actual questions and sort of discussion stuff. I mean, there's quite a few different things that I'd like to briefly touch on before I open this up entirely. I mean, I'm curious, for example, as to what happens if you fully erase this hippocampal engram at 28 days instead of just blocking it, the CA3 to CA1 connections. because I've always learned about, the shift in dependence of the engram from the hippocampus to the prefrontal cortex over time. But now I realize there's a lot more variance that you could be asking about, like, to what degree is it dependent, on what conditions, on the hippocampus or the cortex. What if you silence particular areas? Does something previously it had just been like, do you see memory recall or do you not see memory recall? But now maybe it should be, well, how selective is that memory as well at different time points? Yeah, Randall.
One of the things that I'm curious about is additional phases of long-term memory, because sometimes you cut long-term memory into, say, long-term and long-lasting long-term memory, where you have then additional, say, interleaved involvement of other memories and things like that. And I'm wondering if 28 days, if so, the results that we see, say, for example, in patients where hippocampus is missing or damaged, and then you get that they're not showing the ability to recover this. I mean, you'll see some retrograde amnesia, maybe even for several years, of not being able to retrieve precise memory, but the gist-like memory is not affected. And here, suddenly, there's an emphasis on dentate gyrus and hippocampus being involved in gist-like memory rather than precise memory. It could be that there are still additional mechanisms involved between, say, a 28-day situation and one that is even longer, because in those other cases, the patient cases, they're usually discussing remote memories that are years old.
Yeah. I mean, you can't do that in a mouse, obviously, because they only live about a year. But yes, it would be interesting to see an additional month or two or something, and then the equivalent with human stuff. I mean, I'm also curious about human. Sorry, Alex, I do see your hands raised. I'll get to you in 1 second. But I'm curious about what's happening in humans as well for a few reasons. One is that we know that with humans, there's either reduced levels or no neurogenesis in the human hippocampus. It's still, as far as I'm unclear, as far as I'm aware, unclear as to whether it's just much lower or none at all. But there are also circumstances with humans where you would be kind of ablating this neurogenesis to a degree. So, for example, if cancer patients are being given chemotherapy, you'd expect that to block these newborn neurons. And I'm curious as to, like, do they have effects on their just like memory formation? What's going on under these different circumstances? What are human dentate gyrus neurons up to? But I want to give Alex a chance. Alex, what's your comment or question?
Well, first of all, as a comment, thank you for a great presentation of a great paper. This is my first time here, so this is a lot of fun. I guess the big picture question would be, they sort of orient us to say, look, this is an active gist model, like this neurogenesis happening from CA1 to CA3. What if it's just representational drift and this sort of just passive dependent, like, spillover that I think you mentioned earlier? Do they, I mean, it's a hefty paper methodologically. Do they offer like any, they probably wouldn't be able to like completely reject this idea that this is at least in part just sort of temporal memory degradation in terms of engram.
Just thinking through that. So they're doing the label immediately after the conditioned fear conditioning, the contextual fear conditioning. And they're using those labels that were formed during the initial memory formation to do the imaging at days or weeks later. And they're seeing changes in the structure. They're seeing changes in the pattern of connectivity. But they're not really looking for like other circuitry that has started to become involved in the representation of that memory. I know we've looked at previous papers that have looked at representational drift and the like change in the cells that underlie the engram. But I think insofar as in this paper, they're essentially monitoring the same population of cells, but seeing what changes in them over time. I'm not sure to what degree their results could just be explained by representational drift. Maybe someone else has a thought on that as well. But yeah, that was the. Was there a second part to your question?
Maybe.
No, it's okay.
No, I don't remember, and no pun intended. No, thank you. I get the reasoning. I think I don't know. I'm not skeptical. There's a better way of saying this, but there has to be there's an underlying assumption that this is all goal-oriented, sort of active synaptic restructuring, and it's maybe it's just happening naturally. But I do see your point about immediately imaging it and sort of.
Yeah, no, but I agree with you on the second part there, where I'm curious as to what degree this structural changes are functional, for deliberate sense, versus just selective insofar as the clustered synapses are stronger, so they last longer and they're not just erased over time. The difference though is you saw that increase in density where it went from being 6,000, 7,000 to 20,000 at the remote time points. If it was just going to be selection over time, then I would have expected the density to be higher at the start and decrease over time rather than the other way around. That's my naive thinking, at least. Randall?
Yeah, no, the representational drift thing is definitely a good point. But then, of course, combining that with their behavioral finding, right? Where, for example, they can show that you get the same result of more gist-like if you have more neurogenesis and less gist if you ablate the neurogenesis effect. Those are really interesting. That somehow that is affecting not just some drift in the representation, but also in the behavior. Yeah. And not just that the behavior goes away, not just that you get, say, no response in A and B, but that you can get the same upregulation in B and A. That's kind of funny.
Yeah, I was impressed by the robustness of the results. Let me just run through the last couple of things just because we've got a couple minutes left. Oh yeah, and one other final question I had was about the specificity. So we've seen, for example, in a previous paper, we were looking at the clustering changes in the cortex instead of in the hippocampus, where we're seeing that over time, these sort of procedural memory engrams, these motor memory engrams in the primary motor cortex to dorsal striatum connections change during motor learning. And now I've seen this for the hippocampus, and now I'm just curious about the degree to which this is just a general phenomenon across the brain, which is just kind of interesting. Finally, I want to bring it back to the relevance for memory decoding, which is why we are all here. So just a few points on that I was thinking about while reading the paper, which is just, we've seen another paper showing this clustering change over the course of memory consolidation. And I am just curious about the degree to which these clusters could possibly be used to pick out specific memories, in the sense of is there some way of structurally being able to look at these connections and be able to clearly parcelate connection groups based on their structural connections, at least for memories that have consolidated, is one question. Another question is, can the synaptic density or the synaptic clustering signatures, at least in the hippocampus, be used to date particular memories? I don't know whether that would be useful at all for decoding, but it is interesting from a dating signature perspective. And then one other thing I was curious about is whether there's any sort of degree in variation in the way that the clustering occurs in, let's say, hippocampus versus cortex versus elsewhere in the brain. Because we know that engrams, they're not just in the hippocampus or just in the cortex. You get this different engram formation in different brain regions at the same time. And if you could somehow have the same engram in a behavioral sense, where if you reactivated those cells, you saw specific behavioral activity, but you knew that it was being represented in different formats with different kinds of clustering in different locations, maybe that would somehow be useful for understanding how to do decoding better. But these are all pretty vague comments. I didn't have a specific takeaway of aha, based on this paper, I now know how to do memory decoding, which is, one can hope for, but I feel like I just am, as Randall says, a bit more confused having read this paper rather than less confused about what's going on. And that's it from me. So now we have hit the hour. So if people need to leave, that's totally understandable. But if you want to stick around and chat for a little bit more, we can do that too.
Thank you, Ariel. That was fantastic.
Yeah. Does anyone have any thoughts or confusion, or you can just start now?
Is this directly contradictory to other consolidation stuff? I know that's a big question, just a gut instinct. Is this like just directly contradictory of some other results in the consolidation?
Yeah, I very briefly tried to go hunting to see if this directly contradicted anything, which is why I mentioned that review paper, for example, in the introduction and background. I could not find a clear paper to which this was directly contradictory for my 15 minutes of looking.
What do you mean by contradictory? Do you mean contradictory in terms of, say, the involvement of hippocampus in gist versus precise memory? Or do you mean contradictory in terms of studies that have looked at changes in engrams?
No, I'm saying even from a behavioral standpoint, it's kind of like.
Well, it does seem to contradict some studies there. I did a quick search to see patient studies, and in the patient study literature, it seems that generally, it seems to be the case that where hippocampus is damaged, then you are lacking in retrieval of precise episodic memories, not just like. And just like is still preserved to some degree, although in severe cases you can also have complete retrograde amnesia for episodic memory for some period of years. So this idea that the hippocampus is involved and necessary for the gist-like memory, that is different from what the patient studies seem to show.
Yeah.
Yeah, from what I could briefly find, and again, I haven't done a thorough search on this, it's very much contradictory to previous theories about what was happening in hippocampus and cortex and what the hippocampus's role does. But I couldn't find any immediate behavioral results where, they'd done contextual fear conditioning, and some other study had shown that at 28 days, you could lesion out the hippocampus and still see this specific or just memory sort of activity. I couldn't find any direct comparisons.
But again, there could be a difference in terms of the, how consolidated memories are, because it's 28 days and it's not memories that's years old, as in those patient studies.
Yeah.
I mean, it does seem to be contradictory of the go-to paper, which I have been interpreting as essentially a lesioning of the cortex. It's not really a lesioning of the cortex that they did. They did a kind of a making it not consolidate, essentially. But that got rid of the memory. And this is saying that, and that was 2 days, and this is saying 28 days later, the memory is still there. And the gist is it's still hippocampus dependent. I'm wondering if some of these might be, these seeming contradictions might be explainable by there being two systems that are operating in parallel. And
wait a second. You said, and the gist-like memory is still hippocampus dependent, but that's, maybe I missed a piece here because I was also checking some messages, but did they do a study where they retrieved the gist-like memory or tried to retrieve it when without the hippocampus by deactivating the hippocampus or something? I don't think that was in this paper, right? So you can't, if that's not in here. Sorry, Ariel, go ahead. What was it?
The one thing they do that's relevant to that is they block the CA3 to CA1 connections at the remote time point, and they show a decrease in freezing behavior, but not a complete blockade of it.
Okay.
Which
All right.
is
Well, as long as let's say there's still a way for the consolidated neocortical version of the memory to be retrieved, let's just assume that for a moment, then you can have a situation where you have a gist-like engram being preserved and accessible without hippocampus. And you could have a transition from precise to gist-like in hippocampus at the same time, so that all these studies would hold, and you would still have, and the other studies where you still have gist-like memory without the hippocampus would also still be true. So that's possible.
Yeah, it's just weird that then the under conditions of, let's say, no neurogenesis, the specific one in the hippocampus trumps the gist-likes.
That's really weird. Yep.
But yeah, I guess to answer Ken's question, I'm not sure I've seen a direct contradiction in behavioral results that makes me go, aha, here's a smoking thing for like, it's a methodological issue or discrepancy or something. Cool. Any other questions or comments from anyone else who's come along today? If not, then we've got Ken up in a couple of weeks to look at binary and analog synapses or something. Is that non-hippocampal for once again? We're back to the cortex?
It is indeed. It's cortex.
Woo! That'll be fun. And then I haven't announced this on the chat yet, but in a few weeks' time, we're going to have a discussion of nominating papers for this year's awards and prizes. So anyone who's listening, if you've got any papers that you think would be a good candidate for one of these annual research awards, or you know other people who'd be interested in either having their paper nominated or nominating a paper or anything in this regard, then you should come along to that discussion, or at least nominate a paper on the Aspirational Neurosciences website. But otherwise, I think we can wrap up for today. Thanks for coming, everyone.
Fantastic. Thank you, Ariel. Thanks, everybody.
Thanks. Thanks, everybody.
Bye.
Thanks, everybody.